Welcome to this short presentation of our study group, ESGAI, that stands for ESCMID Study Group for Anaerobic Infection. We will quickly discuss the objective of our study group, some facts about it, and some recent works we achieved. Due to the very nature of anaerobic bacteria and the relative difficulties to work with them, anaerobic bacteria are frequently underrepresented in studies. Even with the spread of metagenomics, this world is still hardly understood. In this study group, we have three major objectives. First, we aim at collecting and spreading knowledge about infection involving anaerobic bacteria. Second, we develop and act the methods used to explore anaerobic bacteria, from culture condition and medium, to identification process, like the development of a MALDI-TOF-specific database, and to antimicrobial susceptibility typing of these isolates. Then, we also explore resistance in anaerobic bacteria, conducting studies on clinically relevant isolates, measuring resistance rates to different antibiotics, and when needed, exploring the mechanisms that are involved. This study group was formerly known as the ESCMID Study Group on Antimicrobial Resistance of Anaerobic Bacteria. That was, as you guess, specialized in resistance to antibiotics within anaerobic bacteria. It was one of the first study groups of ESCMID. The extension of its domain of expertise occurred in 2012. There is, at the time, 67 members in the study group. I will now show you some recent works the study group was associated with. As you may know, EUCAST has recently published a new methods for antimicrobial susceptibility typing for fast-growing anaerobic bacteria. This method uses a new medium called fastidious anaerobe agar and is focused on determining AST in less than 24 hours using disks. Some of ESGAI members worked directly with EUCAST to develop those breakpoints, and the whole group reviewed them, suggesting some changes. Then we evaluated its applicability in multiple laboratories in Europe on both reference strains and local isolates. You can find this method description in the EUCAST website and in the 12th version of the clinical breakpoints. Many members of ESGAI participated in a retrospective study on anaerobic bacteremia that was coordinated by Professor Joan Lambert from France. This study explored a lot of parameters of anaerobic bacteremia, from frequency of isolation of anaerobic bacteria compared to frequent aerobic ones, frequencies of isolation of each species on each site, and evolution of this data between 2012 and 2018. This study will lead to a publication shortly, and it helped underlining the need for future works on some points. ESGAI is also interested in resistance mechanisms. In this study, we explore the heteroresistance of some Bacteroides fragilis strains that we encountered. This study found that a toxin-antitoxin system was implicated in the overexpression of a metallobeta-lactamase, CFIA, leading to increased resistance to carbapenems and the survival of part of the cells. If you are interested in anaerobic bacteria, don't hesitate to apply it through the ESCMID website's application form. We also have a Facebook page called Anaerobes for sharing and discussing news and studies about anaerobes. Don't hesitate to contact us through our mail address, Erin, if you have any questions or inquiries.